Hello, everyone. Hello, everyone. Welcome to the first of our series, How to Create High-Quality Non-Clinical Study Design Based on Clinical Trials. My name is Yukino Ferguson. Thank you for joining us tonight. In this series, we'd like to show you the findings from our own perspective as a non-clinical CRO, what may have contributed to the success in clinical trials, with some examples of companies and compounds that we supported in preclinical stage. As we all know, it is a long journey to develop FDA-approved medicine. According to the data we found, it typically takes 10 years at minimum and a cost on average of $2.6 billion for creating one FDA-approved medicine. As you can see on each step moving forward, drastic elimination occurs. Even the candidates made into phase 1 of clinical trials, less than 12% of that will actually reach to the approval from the FDA. According to the paper published by Nature Reviews Drug Discovery, the #1 cause of failure is the lack of efficacy. Second comes in toxicity issues. Although the pharmaceutical industry has spent tremendous amount of resource and time in drug discovery, the number of incurable diseases remains extremely high. Many promising drug candidates in preclinical stage are unfortunately doomed to be unsuccessful in clinical stage. This is often due to the animal model being used in preclinical research had low clinical correlation, or preclinical studies were designed or conducted very poorly and failing to be effective when tested in human patients. We'll look at the article by Steve Perling, Make Mice Studies Work, published in 2010 Nature. Quote, mice take the blame for one of the most uncomfortable truths in translational research. Even after animal studies suggest that a treatment will be safe and effective, more than 80% of potential therapeutics fail when tested in people. Animal models of disease are frequently condemned as poor predictors of whether an experimental drug can become an effective treatment. Often, though, the real reason is that the preclinical experiments were not rigorously designed, end quote. As he mentioned in the article, there are a huge gap in transitioning from basic research to clinical research. In his word, more than 80% of potential therapeutics fail when tested in people. This drastic elimination seems to be common in every stage of drug discovery process, but more painfully so, especially from preclinical to clinical research, due to the tremendous amount of time and resource has already been invested, not to mention the lives of patients who are counting on the new drugs. This gaps between the basic research to clinical stage is often called the valley of death. Our focus as a preclinical CRO is to bridge this gap by not only offering clinical relevant models, but also creating adequate study designs to increase the number of new drug candidates that will successfully enter the market. One of our strongest pursuit of disease area is fibrosis-derived non-alcoholic steatohepatitis fibrosis. In this particular disease area, we will look closely what is most important factor when it comes to getting approval from FDA. Perhaps there may be some insights in clinical research to create strong preclinical studies. Choosing the right primary endpoint is one of the key factors for designing the relevant preclinical studies. We often overlook what's happening in clinical level. If we look at the list here, we can see the candidate drugs that are being tested in current clinical trials. The endpoint for the new drug candidates are fibrosis and NASH, change in ALT level, and NAFLD activity score, and so on. We can also see what area of targets are being explored by pharmaceutical companies. In this case, FXR and GLP-1 seems to be a popular targets right now. Today we'll talk about the compound targeting galectin-3 inhibitor. Endpoint is histology and fibrosis. And this candidate is currently in phase 3 in clinical trials. In the area of NASH, there are currently 82 different compounds in phase 2 and 3 in clinical trial. As some of you may know, we offer NASH model called STEM model for clients studying in preclinical stage. We have been offering this model over 10 years now. We are finally seeing the results from clinical trials. Here are 5 examples of compounds that are now advancing to phase 2 and 3. These were tested in our model and had positive effects, and now found to be effective in human patients as well. In this series, we'll pick a compound listed here and explore our take on what might have been a contributor for the success in clinical trials. Contents will be shared in LinkedIn website for webinars, so please stay tuned for our upcoming posts. So as today's topic, galectin-3 inhibitor targeting drug, FibroTest score was the endpoint in clinical trials. It is a method of scoring fibrosis by imaging. This medicine was tested for the advanced stage of fibrosis derived from non-alcoholic steatohepatitis. This compound is one of the few candidates that can potentially treat advanced stage of NASH fibrosis. When we designed the efficacy study, we set up our endpoint for fibrosis measured by liver hydroxyproline content and serous red staining. The compound was tested on our STEM model, which has many similar phenotypes as human NASH patients. This is then confirmed to have positive effects for inhibiting the advancement of fibrosis in later stage of NASH. Our focus as a preclinical CRO is to bridge the gap from the preclinical to clinical studies by not only offering clinical relevant model, but also designing the studies that have strong correlation to clinical studies so that we can increase the number of new drug candidates that will successfully enter the market. This one is an example of the successful cases that we were able to contribute, and we hope to hear more favorable results from the upcoming year. So here are a few features of STEM model we used in this study. The pathology includes background of late-stage type 2 diabetes. It also follows same disease progression as human NASH patients, such as NAFLD to NASH fibrosis, and then hepatocellular carcinoma. It also has 100% of mice progress to fibrosis to liver cancer without exception. You can also observe ballooning degeneration of hepatocytes. This is a characteristic of human NASH as well. Also, burnt out NASH effect, the phenomenon in which fat droplets are reduced as fibrosis progresses. Also has a fibrosis develops around the central vein, as well as increased level of human NASH marker, such as cytokine 18. It also has an increased level of human liver cancer marker, glutamine synthase, and also glypican-3 and AFP. They also have a punctual development in each stage. In the study of galectin-3 inhibitor, we chose a treatment regime from 9 to 12 weeks since we wanted to see the therapeutic effect of the candidate drug. Fibrosis is well established by week 9. From here, we'd like to talk a little bit about our model for NASH-related inflammation and fibrosis studies. We'll look at microscopic images and histological HE-stained liver section of our model. In this lowest 2 panel, you can see some examples of ballooning degeneration and inflammatory foci, both of which are characteristics of human NASH and components of the NAFLD activity score. The NAFLD activity score is often used as a primary endpoint in clinical trials, and is calculated by histological evaluation of 3 subscores, which is steatosis, ballooning, and inflammation. Because our model induces these phenotypes, we can use the NAFLD activity score as an endpoint. At 6 weeks of age, our STEM mice have NAFLD activity score of 4 and up, so they meet the criteria for NASH patients. We believe it's necessary to offer both clinical relevant models and also create adequate study designs. This is to show you that our model has the best quality when it comes to testing drug candidates for NASH-related diseases. Here you can see the general parameters. If anyone is interested in the details, we're happy to discuss in a Zoom meeting, so contact us through LinkedIn page or our company website. We offer various biomarkers to evaluate the effect of candidate drugs. Here are 2 examples of biomarkers we use. Cytokine 18 is a candidate biomarker for NASH patients in clinical trials. Serum AFP is one of the biomarkers for hepatocellular carcinoma. We can offer data for various markers to help each client design a relevant study plan. These are more of side note. This slide shows the data for histological staining. On the top panel, oil red staining for evaluating fat deposition. The serous red staining is used for observing fibrosis development. This slide shows some features of HCC with our STEM model. Our STEM model developed the HCC from 16 to 20 weeks of age. Features includes over 4 nodules in any single mouse. It has no visible metastasis. HCC development from NASH is identical to progression in human patients in a short period of time. On the right-hand side, you can also see the pathological findings of HCC in STEM mice at week 20. Here's our survival curve of this model. This shows tested targets and pathologies with this model. Today's topic was galectin-3 as an example for creating preclinical studies with strong clinical correlation. We hope you enjoyed and hope to see you in the next episode. Thank you. If you have a question, we will start the discussion time now. So does anybody have any questions?